Hello everyone, my name is Ray Matulka and I'm the Director of Toxicology here at the Burdock Group. Today I will be discussing some of the critical components and steps needed to evaluate the potential use of novel protein sources for pet food and some of the regulatory considerations that accompany the initial incorporation of new pet food ingredients. First though, I would like to briefly tell those who are not familiar with Burdock Group a little bit about who we are and what we do. Burdock Group is essentially an interdisciplinary team of scientists, regulatory experts, and project managers that help our clients find solutions to complex compliance issues. We primarily focus on food ingredients for humans and animals, including pets, as well as dietary supplements. We work in a brick-and-mortar office based in Orlando, Florida, and have been in business for over 30 years. As with any successful service business, the key to our success is the people that work here. From our founder, Dr. George Burdock, who is a pioneer in the flavor industry, to our most recent team member, Mr. Ryan Parente, who holds a master's degree in nanotechnology and brings practical phytochemistry experience to the group. We pride ourselves on having a breadth of experience to tackle even the most complex regulatory obstacles. You can request more information about Burdock Group and what solutions we might be able to offer you by emailing at infogroup@burdockgroup.com. Also, I do want to state the following disclaimer, obviously for this type of webinar. The concepts and examples discussed in this lecture are for illustrative purposes only. They do not constitute safety or regulatory advice. The examples provided may not apply to your specific situation, so please contact us for further discussion, and we can help you as best we can. So first, I would like to give a quick overview of this webinar. I will be talking first about food ingredient characterization for novel protein-based pet food ingredients, then going into documentation of the source of this new ingredient, then moving into safety of a fermentation or production organism, if that is one of the aspects of its production. I will also discuss intake levels and how intake levels of the specific new protein ingredient are a vital part of the safety process. And finally, if there is not sufficient information, there may be a need for tolerance or safety studies of the novel ingredient in the animal or life stage of animal that are being evaluated. To put this all into context, I do want to discuss a recent market analysis which found that vegan claims stated on pet food saw a 30% compound annual growth rate, and plant-based claims saw a 40% growth rate for the last full 4 years. This is really significant and amount of growth, and the take-home message to this is the fact that there is interest in plant-based food ingredients, and that interest is both for human food as well as for pet food. This will only grow with time. In that discussion, there are different sources of new protein ingredients coming online in the near future or in the past few years. Some of these I've outlined here, such as novel animal sources. One would think that all animal source proteins have already been found and utilized, but there are some novel animal sources, including krill-based products or other animals that are not normally in the pet food supply chain, that are being evaluated for use in pet food. There are also novel or previously underutilized plants that are being evaluated for protein-based ingredients, as well as genetically modified plants. Microalgae has really expanded and grown in its ability to produce protein-based ingredients and other ingredients for humans as well as for animals. Novel bacteria and or yeasts are also being viewed in terms of fermentation processes to produce protein content for animal pet feed. As the same with the plants, genetically modified bacteria are also many times evaluated as being novel, and so they have to be looked upon as a new protein source. We've most likely all heard in the news the aspect of cultured meat and how that is being evaluated. I believe that there are also some cultured meat products have been already consumed by humans commercially. I think that was in Thailand, but that means that cultured meat products may not be far off for use in animal feed as economic feasibilities come into play over time. Synthetically produced protein is also a potential increase in use in animal feed. And by synthetically produced protein, I mean protein produced building block by building block. There is now the ability to produce amino acids, one amino acid component at a time. And in that way, as economies of scale come down and financial aspects come down, these synthetically produced proteins may also be viewed as being utilized in pet food. In discussing characterization of a new ingredient or a novel ingredient, one must analyze the following general points, to which I will go into more detail in the subsequent slides. There must be extensive documentation of the source of the ingredient and documentation of any changes to the ingredient, either from an inherent genetic change in the source organism or changes to their ingredient through the manufacturing process, such as acidification or other manufacturing processes that may modify the protein content. Now, in addition, the manufacturing process itself must utilize any additives that have been found to be safe under the intended conditions of use. One needs to also have a comprehensive understanding of the ingredient, and there must be documentation that the manufacturer can meet the stated specifications, i.e., that the manufacturing process is able to produce the ingredient in a reproducible and consistent manner. So as part of in delving deeper into the characterization of the intended food ingredient, one must have both a general composition, an understanding of the ingredient, i.e. the identity, that includes not just the general understanding of the protein content, fat, carbohydrate, ash contents, nor does it mean only the defining constituents, i.e. what components are included that are the reasons to add the ingredient to food, and knowing that those are in sufficient quantities. But one must have a better and a very comprehensive understanding of the final ingredient, nearly to 100% of what's included in this ingredient. This includes safety parameters and identifying characteristics. So it's not just safety characteristics of heavy metals, arsenic, cadmium, lead, and mercury, but also are the microbials, any total microbial counts in the protein-based ingredient under control and appropriate for the ingredient, but also any microbial contaminants are minimized, such as E. coli or, of course, Salmonella. Any value components, such as specific fatty acids, vitamins, and carbohydrates, must also be a component of the specifications, such that one can show that these specifications and these value components are available in consistent fashion. And also, if there's any concern, like from plants, if this is a plant-based product, are there any residual pesticides or toxins such that analysis needs to be conducted. As I stated earlier, the manufacturing process is also a very critical component to understanding the safety of a novel pet food ingredient. The food manufacturer should have or be able to have access to a diagram of the complete process. And now for plant-based or microbial, or actually for microalgal products, this could mean anywhere from the plant being obtained from a closed or open or wild harvest raw ingredient through the manufacturing process all the way down to packaging. Overall, the documentation for the new ingredient needs to document critical control points, showing that there's a minimal potential for contamination during the processing. Another aspect of the manufacturing process and the overall ingredient itself is to show that the ingredient is stable. It is stable and provides the stated components, the stated proteins or amino acids through the lifetime of the ingredient while it's sitting on a shelf, as well as the lifetime of the ingredient when it's a part of and incorporated into the final pet food product. Now, the components of the stability analysis need to include the components that are the reason for the addition of this ingredient to the food, such as a specific amino acid that you would like to add to, let's say, a cat food diet, overall protein itself. Protein does degrade over time when it's not maintained appropriately. Specific fatty acids may have been included, especially from a microalgal-based product, as microalgae is well known to produce fatty acids as well as proteins, and those fatty acids can degrade over time to provide either an off taste or maybe even it would make the overall ingredient unpalatable to, let's say, a cat or a dog. Also, vitamins also degrade, so we do definitely need to show that the vitamin content that will end up impacting the overall label and label guarantees are stable for the lifetime of the ingredient. Now, protein-based novel food ingredients also will be looking at the protein content and what types of proteins are included. Many plants and microbes produce proteins, and they may produce them in varying concentrations, different ratios, or even just novel proteins in and of themselves. So if you are utilizing this protein-based novel ingredient to replace other proteins like animal-based proteins that are currently utilized in the diet, you really need to show some history of safe use of this new protein, of these new protein components. Or you can show that these new protein ingredients do have substantial similarity to those proteins that are being replaced. And many times you can do that through amino acid analyses or amino acid ratios, showing that they are replacing the animal-based proteins typically for in similar ratios such that it would be an equivalent exchange of the proteins for use in the pet food diets. You can do this through bioinformatics analysis. You would also need to show that these novel or new proteins from novel sources are able to be digestible. This can be done through an in vitro digestibility study. You also need to find out the expression level and dietary intake of these new proteins, such that you don't want to have potential intolerance to these proteins or these accessory components that could be in this protein-based product. As stated here, just because you do a general protein Kaldol analysis or something like that for protein content in this new ingredient does not mean that there could be some components within the protein framework that could be potentially adverse to dogs and cats, such as antifungal proteins or lectins that can actually provide intolerance or adverse effects to dogs or cats. Now, in sourcing the plant protein, you do really, as I stated earlier, you really do want to look at where these are coming from, these new protein products. Are they being upcycled from previous waste streams of plants or plant production? Are the new protein products coming from production organisms, such that an overall biomass is being sequestered and dried and utilized for that from a production of bacteria or yeast related biomass? Or is the new protein ingredient an isolated protein? Is this protein actually coming from, let's say, a well-known rye or other plant-based product in which the protein content is low, but in isolation, you really increase the protein content of the final product? In addition, there is selective breeding processes of many plants and also of microbes, such that you're removing undesirables or keeping desirable components from these plants, as well as there is potential of the ingredient coming from genetically engineered plants or microbes. These all have potential processes in how to and where to find these new ingredients. So in this next part of the discussion is talking about utilizing the machinery of life, such as bacterial or microbial production organisms, to produce biomasses that contain high levels of protein, or then these biomasses have isolated protein content. As I stated earlier, one of the critical components to understanding the safety of protein-based ingredients coming from fermentation processes, either from microalgae or from bacteria or even yeasts, is to understand the fermentation organism. Here are some different questions that you really can look at or you really need to address to a regulator. This is either for the ingredient manufacturer in making sure that you can show safety of the overall final ingredient, or if the pet food manufacturer has questions concerning this ingredient. Here are some questions that you can really ask the manufacturer to make sure that they've really evaluated the product and understanding of how it is being produced. So is the species strain from the fermentation organism well characterized? Is it a native strain, or was it genetically modified? How was it genetically modified? Was it through selective pressure, or was it modified through a CRISPR type analysis? Were any genes inserted or deleted from the fermentation organism? were antibiotic genes, resistance genes utilized? Typically that's not the case anymore, but they still could have been utilized at some point. Was there an upregulation or deletion of native genes? This can actually have a downstream effect on the different proteins being produced. Is there any potential to produce toxins? Are there any toxin-producing genetic sequences in the fermentation organism? As I stated earlier, if you insert any genes or delete any genetic elements, does that change or alter the genetic sequence downstream such that it turns on any toxin-producing genetic sequences such that they are now produced? So, is there any potential for the genus even to contain a pathogenic species? Many times the gene strain or the microbe strain that you're utilizing is not pathogenic, but if you do any genetic modification to it, it could turn on some potential pathogenic elements. So you really have to be cognizant of those concerns as you're looking at these ingredients and you're looking at the fermentation organism as a whole. Because many times in fermentation processes, it's very hard, if impossible, to remove the actual the production organism from the final product. So you'll have hopefully non-viable fermentation organisms or components of that contained within the final product. So the genetic modification, as I stated earlier, is selective breeding to remove undesirables or keep desirable components. Now, many times people nowadays think of this as being a significant concern, or this is something so new and we should be worried about changing the genetics of plants or microbes. But in actuality, as a paper that was published by Dr. Fulton in 1938 shows, it reported that the Hopi Indians in the 1930s and well before that utilized and understood the genetic variability of different cotton plants, leading to different cultivation methods and various tribes having different cotton varieties, indicating that genetic modification of beneficial plants and other organisms have been a part of the human society for centuries around the world, and are going to continue to occur. We just have better technology right now in order to be more definitive in our modifications and to make sure that they are occurring in an appropriate manner and in a safe manner. So in terms of genetic engineering, one must really look at the genetically engineered protein and make sure that it is not a potential toxicant. So you really should look at the following factors. One, what's the source of the gene for the new protein? Is the similarity of the amino acid sequence of interest that of any known toxins? So is this microbial process or plant-based protein able to produce toxins? Is it substantially similar to other toxins that's out there? And is the stability of the resulting protein, is it stable against digestion by pepsin? This can be found or evaluated in an in vitro digestibility assay, i.e. a pepsin digestion assay. Now, when I'm talking about this pepsin digestion assay, this is really just an evaluation if the proteins are readily degraded to small molecular weight fragments via a simulated gastric fluid in an in vitro or tabletop process. This really helps to address the question of whether this protein or any proteins contained within this new plant-based or microbial-based ingredient can be digested and could show if it's tolerated by the pets or a specific life stage of pets. I'll get into more discussion of life stage of pets in a later slide. And as I had stated earlier, throughout a fermentation process and manufacturing process via fermentation of microbial and microbial microorganisms, fermentation may have modified byproducts, creating metabolites that are potentially unsafe for consumers, either human or animal. So many regulators, including the FDA, expects a safety assessment to be conducted on byproducts of the genetically engineered microbes. This can many times be done through bioinformatics or analysis of the protein contents of the proteins contained within the ingredient. Also keep in mind, as I stated, the same with the production organisms, media components must be food grade or safe and suitable for this intended use, because many times those media components cannot be removed from the final product in an efficient or feasible manner. And sometimes in evaluating the different media components, especially for our microbial fermentation, some of those media components may not be readilized in the food supply, so safety assessments may be required. In addition, could there be concerns for worker safety, such as allergenic potential during the manufacturing process, or the use of this ingredient while it's being added into the production of the resulting pet food product? Are there any waste streams from this production that would require an environmental assessment? These are all questions that many pet food manufacturers and pet food ingredient manufacturers should be cognizant of as they're looking at incorporating new plant-based novel ingredients into their overall manufacturing process. Nutrients are known to be essential for growth, but plants, both commonly consumed and novel plants, produce substances to defend against fungi, bacteria, insects, etc. And these components may inhibit nutrient uptake by mammals. So when looking at this, use and production of novel protein-based ingredients, one need to make sure to look at not only if the diet resulting from incorporation of this protein-based ingredient is compliant with nutrition profiles, but as I stated earlier, you need to have a complete understanding of the overall composition of the ingredient, because what is unknown in these ingredients could adversely impact nutrient absorption, leading to intolerance or, at worst, adverse events. So some examples of anti-nutrients include trypsin inhibitors, lectins, tannins. It's well known in pig studies, that some of these trypsin inhibitors or tannins can be found in Phaseolus beans, and if there isn't adequate heating or cooking to significantly reduce these levels, there can be a reduction in ileal digestibility of these proteins when provided to pigs. Now, we don't know how much or if these trypsin inhibitors or tannins affect digestion of proteins when fed to dogs and cats. However, the overall general digestive system of pigs is similar to that of dogs, as well as many different ways of producing pet food products are moving towards minimally processed pet food products and pet feeds, in such that there may not be adequate heating if you would utilize a raw based ingredient from plants that may have high levels of these components. Thiamine antagonists are also different types of proteins that can inhibit thiamine bioavailability, and this could also therefore decrease thiamine availability from the diet, such that the diet in and of itself, when evaluated, may indicate having sufficient thiamine for pets. However, these antagonists may bind thiamine and inhibit its usefulness in the gastrointestinal system, therefore promoting potential issues such as gastric ulcers, impairing glucose absorption, leading to overall decreased feed intake of the pet. As I indicated earlier, the amount of a new ingredient also impacts how to evaluate the safety of the ingredient overall. So one must first determine the estimated daily intake by the amount of this ingredient being added to the feed, and determine if it's a replacement of other nutrients, or will it increase some nutrients over others? Will there be tolerance issues, or will the incorporation of this new ingredient with a high amino acid intake, such as tryptophan or other amino acids, cause amino acid imbalances? Would there be anti-nutritive components from fermentation byproducts, which at low levels have no problem, but when consumed at 10, 15, 20% of the diet, actually adversely affect digestion, again, of other nutrients. So in summary, I just wanted to say, to recap today's webinar, some critical components that one must evaluate when evaluating the usefulness and the potential to incorporate protein-based novel ingredients into pet foods, is that there must be documentation that this new novel ingredient is a safe and consistently produced ingredient, and this is being done through evaluation and understanding the characterization of the protein source, characterization of the final ingredient to be commercialized. Have there been any genetic modifications to the plant and or microbial-based products? Because genetic modification does increase the scrutiny of genetic sequences and any downstream effects that occur in the production of the proteins in these microbial-based products. The viability of the microbe in the final product is also a concern, because if the fermentation microbe is viable in the product, that does increase complexity of the safety evaluation. And finally, if after evaluation of all the analytical and possibly in vitro studies, you still can't determine with reasonable certainty that incorporation of this protein-based novel ingredient is going to be safe for the pet, the companion animal, or the life stage of the companion animal, then you may have to evaluate whether there is a need for safety studies to be conducted to confirm that there is the safety is paramount and shown for this ingredient. So in summary, I would just like to state that the basis for regulatory compliance is safety. And this is safety not only for the intended use of the ingredient as you evaluate it. This can be shown through claims or the safety through the scale-up, as well as specifications can also document the safety of the ingredient for its use in pet food. Thank you very much. I hope you have enjoyed this webinar and hope to speak with you soon.